guess I should have emphasized maybe earlier. All of this that we're going to talk about at first falls under the category of aerobic respiration. And we want to define this very specifically because it has a very specific meaning. It doesn't just mean making energy, but aerobic respiration means the production of ATP from the breakdown of organic molecules. And here's the really important phrase: the production of ATP from the breakdown of organic molecules in the presence of oxygen. If I could write in italics, that would be in italics. In the presence of oxygen. That's what makes it aerobic. Does anyone remember the name of respiration that occurs in the absence of oxygen? Anaerobic respiration. And so we will address anaerobic respiration at the end, but we'll talk about that will be a brief topic. But so far we're going to be assuming that this is in the presence of oxygen, because if oxygen isn't present, only glycolysis takes place. We never get past that. Okay? And sometimes it's desirable to have anaerobic respiration. For example, when you ferment foods, that's anaerobic respiration. So if you like fermented foods or fermented drinks, all of that is anaerobic respiration. Well, not all of that. Most of that is anaerobic respiration. Okay, but we are assuming oxygen is present. Okay, so in the case of our first stage, which is splitting the sugar, glycolysis, we'll say a few little factoids as we go along. This is the only stage that occurs in the cytosol. Remember, the cytosol is the fluid that surrounds all of the organelles, right? It's kind of it's everything inside of the plasma membrane other than the organelles. So it's the only stage that occurs in the cytosol. And maybe just to make the point I'm trying to make, not in the mitochondria. Everything else is going to be taking place in the mitochondria. All right, and glycolysis has 3 phases. So a phase within the phase. So the first one is called the energy investment phase. And if this is new to you, you may be surprised and disappointed when you see this slide that has 10 different reactions when it looked like on our diagram, glycolysis was just one event. Sugar is split into 2 things. That's true, but it takes 10 chemical steps to get there. And so this is a great example of a biochemical pathway. So the investment of energy that occurs at the beginning means that we have to put 2 ATP in, right? The whole point of this respiration business is to make ATP. So this may seem counterproductive. Why do we have to add energy to get energy? But we do. So the energy investment phase, 2 ATP are required in the first steps. We'll just leave it a little vague there. And those ATP are going to do what ATP does, which is contribute a phosphate to something, right? ATPs notoriously have 3 phosphates, one of which is dying to escape and contribute energy. In this case, we're going to put those phosphates somewhere, and of course, again, those phosphates are going to carry some of the energy from the ATP. But this molecule, fructose 1,6-bisphosphate, has those 2 phosphates symmetrically oriented. But before you get to fructose bisphosphate, there's one concept that's associated with glycolysis and the first steps. Actually, I have a picture of the reactions more detailed, I guess. Yes, here we go. So here are the is the first step where ATP is added. We see this glucose 6-phosphate. Now, you may remember, I hope you remember, that we have been using as an example of a coupled reaction the breakdown of ATP coupled with the phosphorylation of glucose is an energetically favorable event because ATP releases energy, adding the phosphate requires energy, but overall it's exergonic. This is the reaction that we were actually talking about when we use that example, okay? And the reason that's important is that when glucose enters the cell, let's see, I'm gonna write some over here. I have more room. When glucose enters the cell, the first thing that happens is that it is phosphorylated, and there's a reason. So this is still under the concept of glycolysis. Glucose enters the cell by tying together something we just learned, facilitated diffusion. Okay, so if there's a higher concentration of glucose outside of the cell than in, glucose will diffuse into the cell, but it needs a transporter protein. And the transporter protein is called, are the transporter proteins, because there's actually a lot of different ones that are in this family. The transporter proteins are called GLUTs. Now it looks like you should call that GLUTs. You do not. You call it GLUTs because it is a shortened form of the words glucose transporters. So capital G, capital A, capital U, capital T, and little s. So the GLUTs are the glucose transporters. And we'll make a little flow chart of the idea of glucose coming into cells later, but just to tie together things that you probably know, the reason we bring glucose into our cells is because we need the energy. The reason that our cells know to bring them in is because we release insulin, right? So the insulin actually causes the glucose transporters to be present in the cell membranes, and then the sugar comes in. So that to tie those two ideas together. Guess I'll erase this, since it's of no value to us over here. Okay, the transporter proteins are called GLUTs, and then when glucose enters, it is phosphorylated and trapped in the cell. Now what we mean by that, or what I mean by that, is that facilitated diffusion relies on transporter proteins. The transporter proteins are highly specific. So if glucose comes through the protein and gets into the cell and then is altered, it can't leave by the door it came in, right? The door it came in only fits glucose, but it's not glucose anymore. Now it's glucose 6-phosphate, which is the first reaction that we're trying to tie together in that diagram. So this is a little trick that the cell plays on itself, I guess, because once it phosphorylates glucose, the glucose is trapped inside the cell. And now, remember, diffusion is about having different concentrations of things across membranes. So if glucose enters the cell and then is no longer glucose, that means the concentration of glucose outside of the cell is still going to be higher. Because every time it seems like glucose is equilibrating the concentration outside and inside the cell, it disappears by becoming something else. It's pretty clever. The cell is clever on many days. Okay. So that's the first step. That's why we're phosphorylating it. Now we're going to harvest those phosphates later and make some energy. But the first thing we need to do is just trap the glucose in the cell. So toss on a phosphate, and now glucose can't leave the way it came in. Okay, let's go back to our picture here. So after we invest some energy into this process, we have a molecule with 2 phosphates. We're going to split that molecule. So we go from 1, 2, 3, 4, 5, 6 carbons. Remember, in organic chemistry, anytime we see a molecule drawn like this, where there's nothing at the junctions, it is implied that there's a carbon there. So in our drawing of fructose 1,6-bisphosphate, we see a carbon written, then there's a carbon at that junction, 2, 3, 4, 5, 6. So 6 carbon molecules split into 2 3-carbon molecules. After we split them into the 2 3-carbon molecules, which to cleave means to split, right? Like you cleave a piece of steak. So in chemistry, when we're splitting things, I almost always call it cleavage of a molecule. So once it's cleaved into 2 molecules, then those 2 molecules go through another series of chemical changes, which include or lead to the energy liberation phase. So we put in 2 phosphates from our 2 ATP, but we end up getting 4. Okay, so we've invested 2 ATP are required, and then in the energy liberation phase, funny way to say it, we yield 4 ATP. And a topic that probably seemed out of place at the time was the idea of substrate level phosphorylation. And the definition of that, we had a slide on it, and the definition of that was when a molecule of phosphate, remember phosphate's a chemical group with a phosphorus and 4 oxygens around it, is when a molecule of phosphate is transferred to another molecule because it was attached by an unstable bond. All right, and so here is the substrate level phosphorylation we have. Where does it say? The removed phosphate is transferred to ADP to make ATP via substrate level phosphorylation. All right, so if we have ADP, that D is di for 2. If we add an inorganic phosphate, we end up with 3 phosphates instead of 2, so now it's triphosphate. So the substrate level phosphorylation is taking a phosphate from this structure where it's attached with an unstable bond, or this structure where it's attached with an unstable bond, and adding it to an ADP. Okay, this is distinct from what we're going to see later in the process. So the reason they emphasize it here is it's different than how we're going to harvest most of the ATP during respiration. So 2 are made there, so a total of 4, because remember our pathway splits into 2 things. And so if we have 4 ATP made minus 2 that had to be invested, our net yield is going to be 2. So if anyone ever asks you how many ATP are made in glycolysis, that's kind of a tricky question because 4 are made, but only 2 are netted. Right? So if somebody ever asks you that, me included, you should ask, are you talking about the net yield or the total yield? Because those are different things. I'm not going to intentionally try and trick you with a question like that. I don't gain anything from tricking you. But I'm just saying, keep in mind, there's a little more under the surface than just the final tally of 2. Okay, let's see what we want to do here. Okay, so after glycolysis happens, again, we've started with 6. We're now down to 2. We have 2 net ATP that have been made. And we also made 2 NADH, which are going to be saved for later. So the NADH came when we had high energy electrons, which are transferred to NAD+. So this is another little reaction to keep in mind. When we have NAD+, and we add electrons, we end up with NADH, which we are going to think of as an electron carrier. It's just a temporary assignment for NADH, so it will eventually give those electrons back to the cell, But the point of making NADH is that it's going to take them from point A to point B and then release them. Most molecules, if we add electrons, we've just permanently altered them, and that's the end of the story. But in this case, we want those electrons to go from one point to another, and then we want to use them again. So that's why we're using electron carriers. Okay. The second stage would probably be a good one to end on because there's really just one or two major steps. So let's go ahead and do this one, and then we'll make a good stopping point for today. All right, so the pyruvate breakdown or breakdown of pyruvate is the second major event, the second of the 4 phases. Okay, and in this second phase, pyruvate is going to be altered so that we take off one carbon to make carbon dioxide. We keep 2 to make acetyl-CoA. Articulate that a little better. Okay, so in pyruvate breakdown, pyruvate, which has 3 carbons, enters the mitochondria. So now we're leaving the cytosol where glycolysis took place. At this point, we will stay in the mitochondria till the very end. Everything else that happens in the mitochondria. So pyruvate with 3 carbons enters, and keep in mind this goes all the way into the matrix. So with the mitochondria, I don't know how much mitochondrial, I don't remember how much mitochondrial anatomy we went into. I know we talked about it having 2 membranes. But if this is the mitochondria, this inner part is called the matrix. And this part between the 2 membranes is called the intermembrane space. Okay, so pyruvate with 3 carbons enters the matrix of the mitochondria. Or if we're thinking of 1, we should use our singular language, mitochondrion. And if you notice, here's something we have learned previously. The idea of 2 molecules moving together in one direction across a membrane requires a symporter. And so as hydrogen crosses into the matrix, pyruvate comes along with it. Okay, so it enters the matrix of the mitochondria. Let's see. And we'll say that it enters via a symporter. Once it's in the mitochondria, there's a massive enzyme complex that acts on it. And the reason we use the word complex is to describe a structure in the cell that has multiple parts or subunits. So if somebody were to describe an enzyme like hexokinase, to my knowledge, hexokinase is a single protein subunit, a rather uncomplex enzyme. But the pyruvate dehydrogenase complex is apparently a massive enzyme composed of many parts. So this just calls it pyruvate dehydrogenase. So I've always heard it called the pyruvate dehydrogenase complex to emphasize its complexity. But nonetheless, let's put that down. So the pyruvate dehydrogenase complex, and anytime we see that ACE, remember, we're talking about an enzyme. The pyruvate dehydrogenase complex, put in parentheses, enzyme, so we remember that, produces acetyl-CoA, right? And the acetyl group has 2 carbons and carbon dioxide, which of course has 1 carbon. So keeping up with the math, we started with a 3-carbon pyruvate. One of those carbons is given away, the other 2 are going to continue this journey. All right, so we produce the acetyl-CoA, carbon dioxide, which diffuses out of the cell, and importantly, another electron carrier. All right, and the acetyl-CoA, a lot of times people just call this coenzyme A, but I don't think your book ever really explains or tells you what that structure is, and maybe you don't want to know. I always wondered what the structure was, and so instead of just calling it coenzyme A, this is what it actually looks like. This isn't something you need to memorize or anything like that. I just wanted you to know, when they say CoA, they're talking about a pretty massive structure that the two little baby carbons in the acetyl group are added to the end of this thing. All right, so this is just kind of a transporter. It's going to house the acetyl group for a short time, but it's a pretty massive thing. And if you pay attention to the structures that we've talked about so far, you might recognize this structure. If we were to add a phosphate to that, bear with me. If we were to do that, take off this, take off that, and add a phosphate, do you know what that would be? It's the main character of our story. It's ATP. Yeah, so why that related molecule is a part of this, I can't say, but I just find that kind of interesting. Okay, so let's call it a day. Let's stop there.